So I wanted to explain protein synthesis in this activity. And I think to be able to understand this activity, we have to understand what's going on during transcription and translation, right? We have learned that genetic information is going to be stored in the nucleus of the cell, right? So, and it never leaves the nucleus. So what's going to happen is that mRNA is going to go into the nucleus, right, of a cell, and it's going to get a transcript of the genetic code to be able to take it to the ribosomes, right? Take it outside to the ribosomes, and the ribosomes will make the proteins, right? A polypeptide, a protein, right? So, this is what you should know, right? And the most essential things that you should keep in mind is that in DNA, we only have 4 nucleotides, and those nucleotides are A, T, C, and G, right? And in a DNA sequence, we will always pair up the A's with the T's, the G's with the C's, and vice versa, right? Simple. That's what happens in DNA. Now, in mRNA, we have the nucleotides A, U, C, and G, right? And remember that RNA is single-stranded, right? So during the process of transcription, whenever we see a nucleotide that has a T, in RNA, we will pair it up with an A, right? So T's with A's. Whenever we see an A, we will pair it up with a U, because in RNA we do not have T's. We do not have T's. All right. And for G, we will pair it up when we see a C, in DNA, we will pair it up with a G. When we see a G, we will pair it up with a C, right? So that's essential that you remember that. So in the nucleus, transcription will happen. So this part of the DNA will be turned into RNA language, and then this mRNA will leave the nucleus with the transcript, and it's gonna take it to a ribosome, and this ribosome will make this polypeptide, right? And it's gonna make that by looking at the code and grouping them in groups of 3, right? Groups of 3, which we call codons. And we are able to determine what those codons are by looking at this chart right here. So, with that information in mind, we're going to start doing this practice, right? And I think it's helpful to be able to write down our cue sheet on the side, right? So I'm going to write RNA, right? And what we have in RNA. In RNA, we have the nucleotides of A, U, C, and G, right? And remember that in DNA, we have the nucleotides of T, A, C, or G, and C. C or G, it doesn't matter. But the reason I've written it this way is to be able to remember that when I'm translating from DNA to RNA, when I see a T, I'm gonna make it into an A, right? And then when I see an A, I'm gonna make it into U. When I see a G, I'm gonna make it into a C. When I see a C, I'm gonna make it into a G. All right, with that information in mind, we're gonna start this activity. Okay, so now I'm given a DNA sequence, so I'm gonna make that into an RNA sequence. So I have a C in the DNA sequence, so if I look at my chart, when I have a C, I write a G in RNA. When I have a T, I write an A. When I have a, I write a U. And I repeat the process, right? T, A, T, A, G, G's are C's, C's are G's, G, C, C, G, T, A, U, C, G, C, G, G, C, A, U. And if I forget at any point, I just refer to my chart. So C is G, T, A, G is C. All right. Now, to be able to make this into amino acids, I just simply start by grouping them in groups of 3, which are called codons, and then I refer to my codon chart right here. This is my codon chart. So GAU, what's GAU? GAU is ASP right here. I'll look at GAU, ASP, right? And then I refer to my key. ASP, what is ASP? Is aspartate, and that's letter D. So I write letter D right here. The same for the next one. AAC, AAC, it's ASN. ASN, I refer to my key. ASN is N. So, write N. Then I move on. GCG. GCG is ALA. That's alanine. That's A. And now I look at AUG. AUG, it's MET. MET is M, the letter M. And then GCU. GCU is ALA. ALA is A. And then GAC. GAC, I refer to my chart. GAC is ASP. ASP is aspartate. D. So now the message we read is DNA mad, right? So and this is a sequence of amino acids. So that's our message. Now for the next one, we are given basically the same code, right? It's the same code as this one, except the T right here has been replaced by an A. So it's basically the same thing. Do the same process, and I'll have the same 3 right here, the same codons right here, same codons, same codons. Same codons. So this will translate to the same thing, D and A, D, and we are just left off with this bit of work to do. So now I turn this into RNA language. AAC AAC. Whenever I have an A in DNA, I have a U in RNA. I look at my chart, A, U. Then A, U. Whenever I have a C, I have a G. Then I look at my chart, UUG is leucine. And then I look at my key, leucine is L. So I write L right here. And now the message reads DNA lad. Now, on the last portion, or the last code that we have to make into amino acids, has an intron. It's telling you that it has an intron. And if you remember from the lectures, we know that introns get ignored, right? Introns don't code for anything. We don't make them into proteins, right? We ignore them. Exons are the ones that we take into account. So now we're gonna look for this portion and we're just gonna cross it out. We just cross it out because it's an intron, and we proceed to doing our translation, right? And if we look at this, they're the same. This right here are the same as this one. It's the same, so be the same. So this one will be a D. This one will be an A. This one will be a D. And now our message reads that. So that's basically all we needed to know.